So this week we're going to talk about complex genetics. So basically complex genetics are all the things besides what we worked on last week. So complex genetics problems, and we'll talk about some examples of diseases. So what are some examples of complex genetics? We'll talk about things like sex-linked traits, meaning that the trait is on a sex chromosome. So this would be on your X or Y chromosome. They're codominants, meaning there's two traits show up together. Incomplete dominance, meaning they blend. And multiple alleles, meaning you have more than one option. This tends to be what's common with hair color. And also this stuff is related. If you plan on taking AP, you'll learn about something called recombination, where the genes mix up, which is what happens with meiosis. So there's some traits where they're linked together, and then people tend to have them, they get inherited together. So these are kind of all the exceptions when people are like, well, what about this? What about a kid who's got one blue eye and one green eye? So first up, we'll start with sex-linked. Sex-linked traits are, again, traits that are on your sex chromosome, so on the X or Y chromosome. Sex-limited means that they're something that you can only have because of your sex. So for example, something like prostate cancer. Prostate cancer would be an example since only men have it, so it's a sex-limited trait. Same thing with penile cancer, uterine cancer. They tend to be reproductive things because only the female or male sex has it. In contrast, something that is sex-influenced is a trait that is influenced because of your trait. For example, things like breast cancer are more common in females, but a male can still get it. The same thing with some of the testosterone-related things. So, for example, male pattern baldness, it's sex influence, baldness is more likely to happen in males because of the levels of testosterone, but you can still get it if you're a female. So how do you do a sex-linked trait? You do it like you would a Punnett square, but you would set it up. Parents on the outside, and you put their gametes on the inside. You'd find that you have a 50% chance of females and 50% chance of males. You'd put the gametes on top. So, for example, if we had somebody who was heterozygous, you could then put the traits as such. And from there, you use similar rules to Punnett squares. So now if we wanted to actually do it all and break it down, we have somebody who's called a carrier. The reason for someone who's a carrier is they're heterozygous, meaning they carry the trait but they don't have it. So this individual, this female, would not have hemophilia. Hemophiliac is a disease where your blood is not able to clot, and you would die, basically bleed death. So in this individual, we have a normal male. Our first female would be normal, big H, big H. Our second female will be a carrier. So we have normal females. %, homozygous, dom, and we have 50% carrier. If we look at the males, it's a different story. One healthy male, one male that's sick. So this male right here is going to be sick. He's going to have hemophilia because he only has that little H, and he can't have anything else rescued from the dad. Again, the Y chromosome is what's making male. Moving on, we can look at codominance. This is when there are two traits together. This is really common with animal color. For example, 2 copies of RR make you red. 2 copies of WW would make you white. So if you put red and white together, that's ruin. You actually have two. You have a red hair next to some white hair, and then red hair. They would show up equal strength next to each other. That's something called rowan. This happens a lot in things like horses, cows. Where else does this show up? In blood types. So in blood types, there's something called the ABO blood type. And the ABO blood type is like this. You could be A and B simultaneously. O is recessive. We have codominance here. And we also have multiple alleles because you could be A, B, or I. So you have 3 alleles here. Blood type is a perfect example of a whole bunch of stuff going on at once. We have codominance, we have regular, dominant, recessive, and we have multiple alleles. So a lot of times people like to use blood type problems to show contrast and to kind of see what happens. It's especially important because with blood type, a lot of people will use this for donations. So me personally, I'm type O. Type O is interesting because it can give to everybody. But the bad thing about O is it can only receive from O. So that's what makes O special. If you're ever an O blood donor, they'll call you a lot because you can give to everybody, and the other ones cannot. For example, A can only give to AB and other A's. B can give to AB. B's can give to B's, but they have some separation there. So in that case, people like to say O is the rarest. It's not rare in terms of number. It's rare as in terms of not a lot of people give their blood. So O it's good. So if you plan on giving blood, this would be a good place to think about. So how do you do a Punnett square for that? Again, you'd write it out. So let's say we had an individual who's heterozygous A, heterozygous B. We write their traits on the outside, their gametes. So remember, these represent kind of their sperm, their egg. What do we get inside? You get an individual who is AB, an individual who is A, an individual who is B, and an individual who is O. So if this ever happened to you, and you were heterozygous A, heterozygous B, you could get every possible phenotype. So if we did our ratios, we would have 1 AB, 1 A, 1 B, 1 O for our phenotypic ratios, and we'd have one of each for our genotypic ratios relating to the genes. So pretty interesting how that could happen. So if you're ever doing a paternity test, you'll realize, hey, you need more than blood type, or the mixed up baby scenario happens a lot. So-and-so mixed up the babies. Can we match up the babies based on blood type? You could go backwards and find that out. The next one we're going to talk about is incomplete dominance. So this is a blending of traits. For example, if 2 R copies make red, 2 white copies make white. If you put them together, you would get pink. Kind of a blending of both white and red. This will happen with things like purple and kind of like blendings. For example, you don't actually see a red pigment and a white simultaneously. You see them blended together. So this is different than codominance. The last one I want to talk about is multiple alleles. So here you just have multiple strengths. For example, black would be greater than gray, greater than white. So you tend to see these a lot in animals, plants, and having a lot to do with coloration. So again, you always are going to go to the strongest of the 3. You need to, again, know the strength of the dominance. So in summary, those are kind of what are talking about. A lot of situations where Mendel's Punnett squares do not apply, especially with humans. Humans are complicated. We're not just peas. Genetics is not a simple input-output system. It's very complicated. We'll get into some of the stuff with biotech and mutations, but the idea is just genetics is not this simple. We do one thing, a bunch of stuff happens after. So remember, lastly, the big point, your genotype determines your phenotype. But you can always overcome your genotype. So your biology is not your destiny. So our biology is not our destiny. You can always overcome. And that's something we'll talk about next week, the idea of nature versus nurture. It's kind of this whole big theme. What really matters? Is it your DNA or is it who you are? So hopefully this lecture was helpful. You got to see some more of the complex examples. If you're really interested in this stuff, please bring it up in class. Genetics is complicated. It's not this simple system. There's a lot of ins and outs and mixes up. So hopefully that was helpful. Again, if you have questions, please comment, call, say something. Don't just sit there like a bump on a log. Cellular aspirating. Have a nice week, and I'll take care. I'll see you around. Bye.